আসসালামু আলাইকুম। আজকে আমরা পড়ব ইসথায়োলজির এক্সক্রিয়েশন চ্যাপ্টারটা। এক্সক্রিয়েশন is the elimination of waste material from the body of an animal to outside, especially the elimination of waste material produced by metabolism. It is the process by which animals themselves get rid of the metabolic waste product and nitrogenous byproduct of metabolism. By excretion, animals regulate their water content, maintain acid-base balance, and control osmotic pressure, the balance between inorganic ion and water. The excretory system promotes homeostasis, the consistency of an organism's internal environment. Vertebrates eliminate some metabolic waste through their gut and the skin integument, but most are eliminated through a special excretory organ, the kidney. Excretory organ and their purposes: integument eliminate urea, some inorganic salt, lungs অথবা gill, make gaseous exchange carbon dioxide and O2. Intestine eliminate some salt, calcium, magnesium. Liver eliminate cholesterol, calcium and ferrous. Kidney eliminate excess water, urea, nitrogenous waste and some salt. Kidney are the main organ for excretion for the of waste product in vertebrate. Structure of kidney: the kidney of the fish are parietal longitudinal structure, lie above the body cavity, ventral to the vertebral column and dorsal aorta, just outside the peritoneum. Commonly they are reddish brown, pulpy and bloody when broken open. Each kidney drains to its excretory through a duct that may fuse casually into a single duct or into an elongated sinus. There are two basic anatomical structures of kidney are recognized in fishes: pronephric and mesonephric. In the pronephric type, anterior funnel lead directly from the body cavity to the pronephric duct by the way of pronephric tube. In the mesonephric type, funnel opening into the body cavity are absent, but instead branches off at the mesonephric duct. The mesonephric tube is possess an enlarged blind in a pronephric tube, pronephric duct, arteriole, glomerulus, nephrostomy, coelom. Pronephros, degrading pronephros, mesonephric duct, coaca, nephrostomy, mesonephric tube, glomerulus, coelom. Degrading mesonephros, metanephros, coaca, male, female, metanephros. এটা হলো প্রোনেফ্রোস, এটা মেসোনেফ্রোস, এটা মেটানেফ্রোস। মেটানেফ্রোস হচ্ছে ভার্টিব্রেটের ক্ষেত্রে, স্পেশালি হচ্ছে টিকটিকি, পাখি, ইঁদুর, মানুষ। আর মেসোনেফ্রোস হচ্ছে ব্যাং, শার্ক। আর প্রোনেফ্রোস হচ্ছে আদি মাছের বা লার্ভার। দিস ব্লাইন্ড in term as Bowen capsule and its draining tubules. Thence they go via the kidney duct toward the exterior. When seen in a section, it shows two main divisions. The outer portion is called cortex. The inner region forming the main mass of the kidney is called medulla. The medulla is composed of several pyramids containing renal tubules projecting into a cavity toward the inner region of kidney called pelvis. Pelvis is the region where the renal artery and vein enter into the kidney. The cortex contains a large number of Malpighian bodies and convoluted tubules. The typical fish kidney is made up of many individual units, each of which consists of Malpighian body, renal corpuscles, and kidney tubules. The kidney tubules join in collecting duct, which finally lead to the outside through the mesonephric duct. The Malpighian body is made up of glomerulus, a blood vessel tightly coiled with afferent and efferent arterioles and encapsulated by Bowman capsule. Glomerular capsule, afferent arteriole, artery, vein, venule, descending line, ascending line, peritubular capsule, renal medulla, renal cortex, distal convoluted tubule, efferent arteriole, proximal convoluted tubule. Difference between kidney and freshwater marine fishes. Freshwater fish kidney or marine water fish kidney. The typical freshwater fish kidney has many glomeruli and comparatively larger. The typical marine water fish kidney less number of glomeruli and also smaller. Blood supply is well developed. Blood supply is not well developed. More water is passes through the kidney. Comparatively less number of water is passed. Kidney weight is comparatively more than that of marine fish kidney in relation to body weight. Kidney weight is comparatively less than that of marine fish kidney in relation to body weight. The number of glomeruli is approximately 10,000. They have fewer glomeruli than freshwater fish kidney. Average diameter of kidney glomerulus is 71 micron. Average diameter of kidney glomerulus is 48 micron. A salmon retained in freshwater has significantly more glomeruli than that in the sea at the same age. A salmon of marine water has less glomeruli than that of in the freshwater at same age. Function of fish kidney. The main function of kidney are elimination of certain non-volatile waste product of the body like urea, sulfate, etc. to regulate pH of blood by eliminating excess non-volatile acid and bases, to remove excess of certain nutrients such as sugar and amino acid when their concentration increase in blood, to remove foreign or injurious substance from blood such as iodide, pigment, drug, bacteria, to maintain the osmotic pressure of blood by regulating the excretion of water and inorganic salt, thus keeping constant the volume of circulation blood, to regulate arterial blood pressure by secreting the hormone renin, to maintain consistency of blood composition, to absorb electrolyte sodium, calcium, potassium, magnesium 2 plus, calcium 2 plus, etc., which are required for fish body, to process the urine formation, removal of certain minor nitrogenous compounds such as creatine, uric acid, ammonia, creatinine, etc. Electric and light organ in fish. The electric organ is an organ that is found in some fishes used to create an electric field. Electric organs are derived from modified muscle or in some cases nerve tissue called electrocyte and have evolved at least 6 times among the elasmobranch and teleost. These fish use their electric discharge for navigation, communication and mating, defense, and in strongly electric fish also for the incompaction of prey. The electric organ of two strongly electric fish are torpedo ray and the electric eel fishes. Location: in most electric fish, the electric organs are oriented to fire along the length of the body, usually lying along the length of the tail and within the fish musculature, as in the elephant-nose fish and other Modimyridia. However, in two marine groups, the stargazer and the torpedo ray, the electric organs are oriented along the dorsoventral up-down axis. In the torpedo ray, the organ is near the pectoral muscle and gill. The stargazer electric organ lie behind the eyes. In the electric catfish, the organs are located just below the skin and encase most of the body like a sheath. Organ structure: electric organs are composed of a stack of specialized cells that generate electricity. These are variously called electrocyte, electroplaques or electroplexes. In some species they are cigar shaped, in other they are flat disc like cell. Electric eel have a stack of several thousand of these cells, each cell producing 0.15 volt. These cells function by pumping sodium and potassium ion across their cell membrane via transport protein, consuming adenosine triphosphate ATP in the process. Post synaptically, electrocytes work like muscle cell, depolarizing with an inflow of sodium ion and repolarizing afterward with an outflow of potassium ion. But electrocytes are much larger and do not contract. They have nicotine acetylcholine receptor. The stock of electrocyte has long been compared to voltaic pile and may even have inspired the 1800 invention of the battery, since the analogy was already noted by Alessandro Volta. In case of electric eel, first detail show electric organ made of stock of electrocyte. Second detail show an individual cell with ion channel and pump through cell membrane. A nerve cell terminal button are releasing neurotransmitter to trigger electric activity. Final detail show coiled protein chain of an ion channel. Electric organ discharge. Electric organ discharge is controlled by medullary command muscle, a nucleus of pacemaker neuron in the brain. Electromotor neuron released acetylcholine to the electrocyte. The electrocyte fire an action potential using their volatile gated sodium channel on one side or in some species on both sides. Electric ray wave হচ্ছে 25 ভোল্ট, স্কেট পালস ভোল্ট হচ্ছে 0.5 ভোল্ট, ইলেকট্রিক ইল পালস ভোল্ট হচ্ছে 600 ভোল্ট।